Okay, welcome everyone to the Zoom and connecting, getting online to YouTube. Hi everybody. Welcome back. All right. Hey everyone, welcome to another episode of Fermentology. I'm Michelle Jewell. You've seen me 100 times by now, hopefully. But I am the science communicator here based in North Carolina at NC State at the Department of Applied Ecology. And this is Fermentology. It's our semi-weekly-ish live stream and a mini seminar series on the culture, science, history of your everyday fermented foods. We've had guests from all over the planet join us, and we're so excited today because we're going to be learning about experimental novel misos with Joshua Evans. And Joshua is a PhD candidate at the University of Oxford, though he's also affiliated with the University of Copenhagen and did a lot of research there for this project. Now we will, as always, have a live Q&A at the end, but I won't be able to get to everybody's questions, so I apologize for that, but please put your questions in the chat as you have them. Without any further ado, Joshua, if you want to go ahead and share your screen and get us going.
Thanks. Let's see here. Is it working?
Yep, looks great.
Great, thanks. All right, so hello everybody. Thanks so much for joining tonight. It's really a pleasure to be part of this fantastic series. If you have been following along since early on, that's really great. If you've joined more recently, I can really recommend going back into the archive of talks. Michelle and I were talking a little bit before we started tonight about how all of the talks are still available and are just as relevant as they were, and they're a really great resource. So tonight, well, I guess it's night for me, but for many of you who are in North America, it will be afternoon. But today, I wanted to tell you a little bit about my current research. So I'm researching microbes and humans and how they change each other in novel fermentation practices. So I'm going to describe a little bit first about how I arrived at this research, tell you a bit about one experiment that I've done for my PhD, share some of the results, and finally reflect a little bit on what these results might mean. So as this series of seminars is all about fermentation, I suspect that many of you will probably already know a little bit about Miso and what it is and how it works, but I'm going to start with the very basics just to make sure we're all on the same page. So if you don't really know much about Miso, don't worry. We're going to start from square one. So, but first, I want to tell a little bit about how I came to this, this kind of research. So it really began at a place called Nordic Food Lab in Copenhagen. This photo was taken there when I was doing research there. It doesn't exist anymore, but it was a nonprofit organization that was set up by a restaurant called Noma. Maybe some of you have heard of this restaurant. It's become relatively renowned in the last decade or so for the approach that it's taken to rediscovering and really championing a lot of these Nordic ingredients and foodways that either had been forgotten about or had been never really kind of developed in a gastronomic way. And so the team at Noma set up this place called Nordic Food Lab to investigate the edible potential of the Nordic region and to share these results openly in an open source way. So we worked a lot with neglected and underutilized foods like wild plants, seaweeds, insects, and also a lot of fermentation. And the approach that we were pursuing involved combining different techniques from around the world with ingredients from the Nordic region to create flavors that were reminiscent of the traditional product, but also flavors that never really existed before. By now, this approach has become fairly common in restaurants and even many home kitchens around the world. At that time, it was maybe a bit less so. So it's great, it's great to see that that so much has changed in the last, 5 or 10 years, not just in fermentation, but in cooking generally. One sort of really good example of this technique that I hope really illustrates it was one of the first experiments that Noma developed called Piso, what they kind of came colloquially to call Piso, which was a miso that was made using, instead of using soybeans, which would be traditional in Japan, it was made using yellow peas. We can almost think of this as a kind of translation, as if we were translating a book from Japanese into Danish, for example. Now, in all of these fermentation experiments, flavor was the main goal for these chefs. But as I learned more while I was at Nordic Food Lab about the microbes themselves that were involved in these processes, how quickly they reproduce, how quickly they can adapt to new environments, the more it became clear to me that these experiments in translating fermentation techniques might not only be creating new flavors, but they might be having bigger effects. They might be also shaping microbial life without these chefs realizing it or willing it. And so that idea became the seed of my subsequent PhD research. So I really wanted to investigate whether the pursuit of these new flavors in these novel fermentations might also be changing the microbes and the humans involved, and if so, how? So to do this, my project has combined fermentation experiments, DNA sequencing, and ethnography to get at how these humans and microbes are shaping each other in what feminist science studies scholar Donna Haraway calls a dance of mutual becoming, which I find a really kind of apt phrase for this process. While the ethnography, so doing sort of anthropological methods, really forms a big part of my overall project and the thesis, in this talk, I'm going to specifically focus on the experiments that I did and the DNA data. So here are kind of the overview of 3 experiments that I did. One was with miso, and this was at Noma to see if making misos with different ingredients changes their ecology. The second was with koji. Now, koji is a fungus that is used to make miso. We'll talk about that in a minute. And I was looking at whether growing koji over and over again with its own spores, sort of like saving the seed from a plant and then using it to plant the next batch, if growing it this way at different sites, at Noma and at a distillery that was founded by 2 former Noma employees called Empirical Spirits, and in the laboratory at the Danish Natural History Museum, if growing it at these 3 sites would lead to these different populations becoming different from each other. They would evolve differently. And the third experiment was with kombucha, made by a colleague of mine at another restaurant in Copenhagen called Amas. And this was to see whether kombuchas that he had made by assembling them from scratch were different from kombuchas grown in a more traditional way by using a mother. All of these experiments were based on or study existing practices of novel fermenters in Copenhagen. But in this talk, I'm going to focus only on the first one, because we only have time for one. And this is about misos. So firstly, let's talk about what a miso is. What is it? It's a traditional fermented product from Japan. Many of you will know this. And it usually combines cooked soybeans and this fungus called koji that's usually grown on a different grain, often rice, but sometimes barley or wheat, and mixing those with salt. And then this kind of mixture, kind of like a paste, is fermented for a few months or sometimes a few years. And it yields a really umami-rich paste that can be used in all sorts of preparations, from soups and sauces to glazes and marinades. I'm sure that a lot of you are probably familiar with miso. Maybe a lot of you cook with it yourself in your kitchen. But how does this miso, go from being kind of like a slightly unappetizing mash of beans and grains to being this delicious, amazing, umami-rich paste? Well, a lot of it has to do with this koji, this fungus. Its scientific name is Aspergillus oryzae. And what this fungus does is it makes a lot of enzymes. These enzymes break large molecules found in foods down into smaller molecules. So they turn proteins into amino acids. they turn starches into sugars and fats into fatty acid chains. And in the context of miso, they really help to create the amino acids that produce this umami taste. They also, a lot of the sugars from the koji help bacteria and yeasts to grow in the miso, which produce acidity and produce other flavors. And that's really where it's getting a lot of this rich flavor from. Miso also has analogs in Korea and China, and it's found in a variety of forms throughout Japan. Some of them are more sweet, some are more savory, some are more salty, some are less salty, some are very young, some are much older. There's hundreds of different kinds. So in this experiment that I conducted, it was based on Noma's standard recipe, which had been developed over 8 or so years. This involves 3 parts of bean to 2 parts koji, and then 4% of that total weight in salt. It's quite standard ratio, except the salt is very low compared to traditional misos. And we made 8 different kinds of miso by varying this proteinous substrate. So instead of using soybeans, we used yellow peas, we used fava beans, we used a specific type of lentil from an island in Sweden called Gotland. These lentils are called Gotland lentils. And we also cooked each of these 3 substrates normally in water, and also we nixtamalized them. Now, nixtamalization is a technique from Mexico and Central America that's traditionally used to process corn. Many corns, if you just boil them in water, they're still very tough, and it's kind of impossible to make a dough out of them. So if you want to make masa out of it, which is what you need to make tortillas and tamales and all of these different amazing Mexican preparations with corn, you have to nixtamalize it, cooking it in an alkaline solution, so in a solution that has high pH. Traditionally, this might have been done with ash from a fire or calcium hydroxide. Nowadays, it's mainly done with chemicals like calcium hydroxide that are found naturally. And what this process does is it breaks, similarly kind of to the enzymes in the koji, it breaks down some of the molecules that make it possible to emulsify it into a paste. So this is a really good example of techniques that Noma had already been mixing in this process of making translated fermentations. So not just drawing on Japanese traditions and bringing them to Denmark, but mixing, in this case, a Japanese technique with a Mexican technique to yield a fermented product that probably doesn't really have any kind of precedent before. So we nixtamalized them. And we also made misos with soybean as a kind of control, like a normal kind of traditional miso, and one with rye bread. Rye bread is a super Danish staple that people eat, all the time. It's also rich in protein. You can make miso out of it. So we made each of these 8 misos in 3 replicates, so making 3 kind of batches of the same miso. The reason for this was to ensure that we had consistent data across batches. Often, many of you will probably also know from experience fermenting that that even if you use the same recipe with the same ingredients, sometimes for some reason it can just turn out very differently. And this is because sometimes even very, very small differences in the starting conditions of a fermentation can yield very different, can yield bigger differences further down the road. So by having them in 3 batches, we could kind of control for this possible batch variation. We fermented everything for 3 months and sampled them at the start and in the middle and at the end. And then so this is a photo of the misos when they're fermenting in the Noma fermentation lab. They were fermented in a room held at 30 degrees, I believe, and I think 50 to 60% humidity, maybe 70. This is a photo of all the finished misos after they had been sampled and packed down. You can even see there's already some differences in the color and texture according to the substrates. And once we sampled them, I brought those samples into the lab and extracted the DNA, purified the DNA, prepared it for sequencing, and then had them sequenced for their DNA. And this was the result. Now, we don't really have time to go into great detail reading this data, but I just wanted to show you this to give you a sense of what some of the data analysis that we can do with this data looks like. Along with the samples from NOMA, from the NOMA experiment, I also got some samples from 6 misos that were made from the same recipe in Japan. These were made at a restaurant in Tokyo called Inua that was affiliated with NOMA. It was founded by a former R&D chef that used to work at NOMA. Sadly, it had to close last year because of COVID, but I managed to get these samples before that happened, which was really great for my experiment. And even though there's no batch variation with these, there's only one sample per type, we can still use them to compare and get some useful findings compared to the NOMA misos. So without further ado, let's talk about some of the results. Okay, so we have some pretty interesting findings that we can conclude from this data overall. So the first one is that the miso ecologies do in fact vary according to their ingredients, but interestingly, some vary more than others. For example, the fava and soy misos had a very clear effect, whereas the pea and lentil misos had some effect, but it was a bit less clear. And there are a few reasons why this could be. The second result is that there was a really clear effect of the nixtamalization process. So the 3 nixtamalized misos that we made had a very similar ecology to each other, even though the 3 ingredients that they were made from differed. So this is very notable. Third result that we can find from this data is when we compare the piso made in Copenhagen and the piso made in Japan with the same yellow peas and the same barley, had comparable compositions, but the compositions were in different proportions. And that sort of has some interesting interactions with the previous 2 findings, which we'll discuss in a moment. So furthermore, these novel misos exhibit overall more diverse ecologies than standard misos. And I say standard in quotes because this is comparing this data to misos that are described in the scientific literature. Now, I've been searching for any kind of scientific paper on miso ecology for many months, and I've only really kind of come up with 1 or 2, and these are quite old papers that don't use sequencing techniques, they use culturing. And so I was very surprised at this. There must be some papers that study miso ecology in Japan, possibly in Japanese. But at least it seems so far in English, there's very, very little on this sort of thing. So hopefully as more studies come out, we'll have more things to compare it to. But so far, it seems that somewhat unsurprisingly, these novel misos that are really mixing and matching different ingredients have more diverse ecologies. All right. And then finally, give me one moment. There we go. All of these findings we can kind of sum up by saying that taken together, they strongly suggest that treatment, which is to say how one makes the miso, tends to matter more than geography, i.e. where the miso happens to be made. And this is in contrast to a lot of previous studies, which I'll go into deeper in a little bit. It should also be said, though, that these data only go to the genus level, and there are probably some more important differences happening at the species and even strain level that this data so far isn't detailed enough to pick up on. So that's something I really hope to do in the future. So why does this matter, aside from the fact that miso ecology is fascinating and everyone should just intrinsically care about it? One way to answer this question is to see how these findings inform the concept of microbial terroir. Now, some of you might have heard of this idea. It's an idea that became common in the food world around 2012 or so, as fermentation was growing in popularity. It draws on the French notion of terroir, in which specific food products emerge from within specific geographic regions. And so this idea of microbial terroir kind of imagines that specific fermented products are shaped by a unique microbiome associated with where they're made. This seems quite, intuitively true. Often this idea of microbial terroir, at least in English, can imply that terroir is naturally occurring, that it exists prior to people, and that it doesn't change over time. But a more complex understanding of microbial terroir, however, has since been emerging more recently. A well-known study of microbial ecology of cheese rinds, for example, by a microbiologist named Ben Wolf and his colleagues, some of you might know of him or even know him, found that the main factor contributing to different community compositions in these cheese rinds was not geographic location, but was actually specific human practices. So for example, how much salt was added to the cheese, how the curd was cut initially, and how the cheese was treated during aging. Other studies have similarly illustrated how different practices and arrangements of space and food production can shape this microbial terroir, such as in wineries and breweries and bakeries. One recent example is a quite big study of 500 sourdough starters that was just published, I think maybe this week even it came out officially, by the Dunn Lab at North Carolina State, which is the same group that's hosting Fermentology. So if you're really interested in digging more into that, I can recommend that you check out that paper. It's really fascinating. This Miso experiment that we've talked about today contributes to this emerging field of research by showing how human interventions that are oriented towards producing flavor have effects on the microbiomes of the Misos. These data add to this newer understanding of microbial terroir, suggesting that it is not something that's prior to and unchanged by humans, nor even something that we've been participating in and shaping for thousands of years, but that it's really something that, that we're continuing to shape all the time throughout our ongoing practices, whether or not we're trying to, whether or not we're realizing it. In other words, we are already always part of our microbial terroir. Now, to sum up and conclude, there are plenty of directions for further research I think it would be really fascinating to explore. One thing that we could do is produce replicates of the Japanese Misos to control for possible batch variation, and that would make the comparison much more robust between the Japanese Misos and the Copenhagen Misos. Another one, which I would really like to do, it just takes a bit more money, is doing deeper sequencing on these samples to try and resolve the microbiomes more specifically. So trying to gather data on the species and even particular strains to see if these more detailed data suggest new or different conclusions. Together with a colleague of mine, actually, at MIT Media Lab, I've already created another extension of this Miso experiment that I wanted to share with you a little bit, because we are in the process of generating some data on it probably next week. So last year, together with my colleague Maggie, we sent a sample of soybean miso up to the International Space Station to ferment for 1 month. And this photo here is a very zoomed in, very grainy iPhone photo taken from Florida, I believe, of the rocket launching up to the ISS with our little pot of miso on it. And we did this because we wanted to see how the environment of space in microgravity and being constantly bombarded with cosmic radiation might affect the miso ecology compared with earthbound samples made using the same recipe. So our lab work, we got the sample back, we sent it up to space last spring in March and April. We got it back. Everything was shut down because of COVID, so our lab work has been a bit delayed. But we finally have the samples, and next week we're going to start preparing them for sequencing and generating some data on this. And hopefully, if all goes well, we'll be able to compare the space miso with some of the existing misos to see what difference space makes. In general, this comparison, I think, hopefully can generate some further insights into how in fermentation, and especially in these novel fermentation practices that I've been describing, we're constantly participating in this dance of mutual becoming that Haraway talks about, being shaped and shaping the microbial world, whether we know it or will it or not. And with that, I would like to give some thanks. Big thanks to the Noma Fermentation Lab team for hosting me and for helping me in this experiment, to my lab group here at the University of Copenhagen, the section for evolutionary genomics, for helping me with all the lab work and the sequencing. I have a great colleague, he's a biogeographer named Flora Mazel in Switzerland, who has been helping me with all of the analysis and the number crunching. And of course, big thanks to Fermentology for inviting me to speak. It's such a pleasure to be part of this great series. And hopefully now we have some time for questions.
Thank you so much, Joshua.
Yeah.
We have loads of questions. Buckle up.
Great, bring it on.
Here they come. Well, first off, I'll start with a plug as well. The first thing I wanted to mention was that Benjamin Wolf gave a fermentology talk. This was back in June last year. So if anybody wants to check out Ben's talk as well about cheese rinds, and his title of his talk was The Microbial War and Peace in Cheese Rind Microbiomes. And that was a super fun talk as well. So, and also you mentioned to also plug the paper that's been published from the Dunn Lab. That came out, oh my gosh, I'm losing space and time. I don't even know what day of the week it is anymore, but I believe it was earlier this week, to be honest, or maybe last week. I'll throw that link in the chat too, for those of you who want to read, and really great summary that was written by my colleague, Matt Shipman here at NC State. And let me just put it into the YouTube chat as well. So, nixtamalization, which is a phrase that I have just learned today. There are several questions about it, and I'm going to start with Natalie. The technique, it was developed in the Americas. When was that knowledge brought over to Japan for miso creation?
Okay.
Is it standard in miso recipes as well?
Right. So thanks for asking this. Maybe I didn't clarify this enough, because I kind of was just like, okay, we're talking about miso, and suddenly we're talking about nixtamalization together. And so they, as far as I know, I mean, Japan has done it, but as far as I'm aware, nixtamalization does not feature in traditional Japanese miso making. It's an entirely separate tradition. And this is partly what's quite novel about some of the novel misos that Noma has been making over the last few years, is that they're sort of bringing these techniques and these traditions together and saying, well, if we've already been doing some nixtamalizing of different things to make certain flavors, and we're making misos, what happens if we put those together?
And Jeremy was curious as well with that. Is the effect of the process that it increases enzymatic activity?
Good question. So one thing that was very clear in the experiment that I ran was that all of the nixtamalized misos. So firstly, it should be said that, of course, long before I could generate the sequencing, the DNA data, I was already interacting with the misos sensorily. So, over the experiment, I was taking measurements of temperature and humidity and pH also, and also especially noticing, how it changed its texture and its smell and its taste. And one thing that became very clear about all the nixtamalized misos was that they became noticeably more moist than the other misos, even though none of them had water or so added to them. And all the grain, all the legumes and things were cooked in a quite standard way. But all of the nixtamalized ones were wetter than their non-nixtamalized counterparts. And that makes sense in a lot of ways. The nixtamalization doesn't necessarily produce more enzymes. That's mainly what the koji is doing. But then the nixtamalization is kind of physically and chemically breaking down a lot of the larger structures in the pea, for example. And so what that means is that it sort of breaks the structure down more than a non-nixtamalized one, and breaks some of the molecules into smaller pieces. And so when like other microbes in the miso start to grow, like bacteria, for example, then it gives it makes more of its molecules more accessible to these microbes, and so it sort of expedites the process of breakdown. And so that's probably why they were also kind of looser and softer, and it also made the flavors kind of converge. Actually, the flavors between the pea and the lentil and the fava bean, the nixtamalized ones were kind of each of them less distinct in flavor. They kind of shared, well, if you know the smell of sort of proper like Mexican corn tortillas, almost this smell actually mainly comes from the nixtamalizing, not from the corn. And so when you do nixtamalization on other things, you often get like a similar kind of slightly ammonia sort of note.
Nice. Yeah, we had a ton of questions about what sensory differences happened with these misos as well. And do you have any that were your favorite that you were tasting, or if you had to rank them top 3, what would they be?
Right. So, I mean, I really mainly talked about the ecology today, but in the context of the whole project that I'm doing, of course, taste is kind of the central or very important idea, because when it comes to the teams at Noma and Empirical and these other kitchens that I've been studying, when they make ferments, of course they're not sequencing the DNA to find out what's in it. They're tasting and smelling it and watching it, and just like all of us when we do fermentations at home, But I think what became really interesting is that when some of these team members are doing it, especially if they have a little basic knowledge of science, they're able to kind of match up some of the sensory impressions with some knowledge principles of science to sort of be able to infer and say, okay, well, this miso is getting a lot of fruity notes. And I know that these fruity notes are esters that are often produced, when certain yeasts are interacting with certain bacteria or whatever. So I can kind of know implicitly that this miso likely has more of this yeast than this other miso or whatever. And that sort of thing, I think, became very interesting in the course of doing the ethnography. That's a whole other story that we won't talk about. But your question about, did I have preferences or so? I was really interested in, so not just tasting them myself, but also tasting these misos with team members at Noma in the fermentation lab and saying, okay, both, how would you describe this? And also, do you like it? Or which ones do you like more? Or which ones do you like less? And that sort of helps to paint this larger picture, not just of how these processes might be changing the ecologies, but how when certain misos are selected for over others, it helps to propagate certain ecologies forward into the future and let other ecologies kind of die off or not be propagated further. And that, I think, is putting those two sets of data together is where things start to get really interesting. Whether there were ones, I would say, like the nixtamalized ones were actually surprisingly less delicious than we were hoping for. Some of the flavors kind of got a little bit musty or kind of indistinct. It was also interesting to do the soybean control as part of the experiment, because usually, we were doing all of these kind of novel things and not really, it was sort of fun to do a really traditional one next to them, because it reminded us of how delicious soybean and rice koji miso is. There's a reason, I think, why, all of these East Asian cultures were using soybean and all sorts of varieties of soybean. I mean, it's delicious. You can't get around it, really. So.
And last question I'm going to pick from here. There were so many questions, and let me just tell everybody, Joshua is on Twitter, so if I don't get
Instagram.
And Instagram.
Yeah.
If I don't get your questions here, which I won't, please find them there as well for to direct these questions to. But quite a few questions came in about your space experiment. And
great.
Is it kind of the same setup? Were you sending loads of different samples? Talk more about that.
Yeah, so the space miso. Well, as you can imagine, it's quite difficult to send something up to the ISS. It takes a lot of time, a lot of bureaucracy,
Mhm.
a lot of money, which was not my money. This came from MIT, which was great to have a collaborator there. And also space is very limited, and so I think we had space for one miso pot, and it was a lot smaller than the average. I mean, the batches that I made at Noma, for example, were each, I think, I can't remember. I think 2 kilos each, which is also not that big, but it's enough for it to ferment properly. Whereas the space one was maybe, yeah, 200 grams or so, 250 grams. So it's, and that's the main thing where it's not perfectly comparable, mainly because even if the recipe is the same, the difference in volume can actually make a big difference for how the fermentation proceeds.
Sure.
But we're hoping that it will still yield some interesting data. From a sensory point of view, it tastes like a miso. We did a sensory analysis of it, and it's still a miso. But we sent that little miso up onto the ISS in a box that was monitoring a lot of environmental data. So it was recording temperature, humidity, pressure, off-gassing, and radiation. I'm particularly excited about radiation, actually, because at least from principle, that should be maybe one of the factors that's different from between space and Earth, that could be maybe shaping the ecology differently. But only the data will tell. And it was up there for 30 days, and then it flew down, plopped into the Pacific Ocean, and boat went and got it and brought it back, and then we had it in the freezer, and now we're gonna sequence it.
Are you gonna taste it?
Oh yeah, we did. So we did a sensory analysis last week in the lab, and we had two controls. So we had one that I fermented here in Copenhagen, and one that my colleague Maggie fermented in Cambridge, Massachusetts, and the space one. And we did a little, together with a couple colleagues from the lab here in Copenhagen, we did a little sort of double-blind sensory analysis setup. And the space miso, well, firstly, I should say the space miso definitely tastes different to the other two. But this is also probably partly because the space miso has traveled a lot more than the other two, and it's been jostled around a little bit more. And of course, that isn't the same as having been to space. So there's also a little bit of a caveat with this whole experiment, that it's very much an exploratory experiment. There are still some confounds that are built into just the kind of logistics, the logistical difficulties of getting something up there and back. But there's still some space for it to yield some very interesting findings, I think. And hopefully maybe in the future, we can do more. It's part of my colleagues' research, part of a larger project that is looking at sort of trying to think about near futures in sort of futures around eating in space that will happen, in not too long. Not just from a kind of functional point of view, which is how most of this research tends to happen. Like, are astronauts getting enough nutrition, and can they eat things that don't leak everywhere and destroy the space shuttle or whatever? But astronauts who are up there for months at a time, also many of them say that the hardest thing is the monotony of the food and how much it sucks, honestly. And so there's a lot more work being put into, can we make foods that are delicious, that are satisfying to astronauts, especially because when you're up there, your senses are really dulled and you taste things differently. And so part of what my colleague and I are trying to develop is a kind of way of approaching, perhaps in the not too distant future, we can create ways for astronauts to not only eat fermented foods in space, but to do fermentations themselves in space, and then eat these foods that hopefully can be more delicious and more, maybe even have specific flavors that are a result of where they are. And that could maybe also not just give them a bit of, gastronomic satisfaction, but could give them a sense of like emotional connection to their food production, and having a little piece of earth, that ties them to this larger web of life. That's, I think, something that would be very satisfying to a lot of the people who are doing work up there.
I am so looking forward to whoever makes the first sourdough starter on Mars or something.
Yeah.
Like that is going to be a really fascinating study in and of itself. But Joshua, thank you so much. I'm going to quickly plug the next talk that's coming up in 2 weeks. If I can get all my things out of the way. We're jumping straight into wines and cheeses. We have a duo joining us on February 11th, which is in 2 weeks' time. And that's Charles Ludington, who's a historian of both wine and cheese, and then cognitive scientist Anne Sophia Barwich. And together they're going to walk us through a tasting history of wine and cheese. So I'm not here to tell you that it's a great excuse for a cheese board. I'm just saying, if you were to bring your own wine and cheese along, it's probably, whatever time it is in the world for you right now, it's probably a good excuse to do it. Joshua, thank you again so much. Again, find Joshua on Twitter or Instagram. And thank you so much for your time again. Really appreciate it.
Thanks for having me.